major histocompatible complex. We use it to say major histocompatible complex. These major histocompatible complex molecules are present on the cell surface, which are unique to each and every individual person. That major histocompatible complex molecules are unique for each and every individual person, because these are actually from the inheritance process. It's a polygenic inheritance, we use it to say. That means the multiple number of genes are going to be responsible for these particular molecules. So that means every person will get some number of genes from mother and some number of genes from their father. So that's why every person molecules are different from one person to the another person. So we classified the MHC molecules into MHC class 1 and MHC class 2. MHC class 1 molecule. MHC class 1 molecules are present in all the nucleated cells. The cells which contain nuclei, they definitely have this kind of molecule. That way we learn. Okay, so we're doing it back again, no problem. That those who are not seen this, so for them, I'm revising MHC class 1, all nucleated cells, present in all nucleated cells. When a microbe is infected, that microbe infects the host cell, that means our body cell. At the time, the cell is called as altered self cell. Altered self cell. Infected host cell can be called as altered self cell. A nucleated cell is having MHC class 1, and when it is infected, that all nucleated cell, any nucleated cell, when it is infected, that we say it as altered self cell. So to whom it is going to present the microbe, that means we say that T lymphocyte. T lymphocyte, particularly T cytotoxic cells, which leads to that cell-mediated immunity, which leads to cell-mediated immunity. The T cytotoxic cell leads to cell-mediated immunity. Okay. So before we go into the cell-mediated immunity, that we need to remember this. So MHC class 1 molecules are present in all nucleated cells. When they are infected, we call them as altered self cell. Along with the microbe, cell will be presented to the T cytotoxic cell, which leads to cell-mediated immunity. MHC class 1. MHC class 1 molecules are present in antigen-presenting cells. MHC class 1 molecules are present in antigen-presenting cells, APCs. We say that generally dendritic cells, macrophages, dendritic cells, macrophages, so B cells are said to be that antigen presenting cells. Okay. So these antigen presenting cells going to present the antigen to T helper cell, a special kind of cells. We say T helper cell. These T helper cell will make two possibilities. Either they may present it to the T cytotoxic cells, either they may present antigen to the T cytotoxic cell, or they may present the antigen for killing to the B lymphocyte. If it all sent to the T cytotoxic cell, that leads to cell-mediated immunity again. That leads to cell-mediated immunity again. Then if it is given, the T helper cells are given to the T lymphocytes. If it has given to the T cytotoxic cell, which leads to cell-mediated immunity. Wait. So here it is again. So that means for cell-mediated immunity, there are two possibilities. Either from MHC class 1 or MHC class 2. either by Mhc class 1 or Mhc class 2. Okay. So then need to discuss about this today, that cell mediated immunity and humoral immunity. So that's it. Okay, so let me represent you once again that possibilities of the cell mediated immunity separately. Cell mediated immunity. Okay, so the immunity which is possible by T cytotoxic cells is called cell mediated immunity. So how it is possible? It may be by MHC class 1 molecule containing cells, MHC class 1 containing molecules, or it may be MHC class 2. Okay, so that the sequence we need to remember that make us to learn easily the topic here. MHC class 1. So where do you see the MHC class 1? In all nucleated cells, when they are infected, we call them as altered cell. They present the microbe to the T cytotoxic cells, okay, which leads to cell mediated immunity. MHC class 2 is present particularly in the APC, and from the APC, the dendritic cell or macrophages or B cell, whatever, we call them as APCs, and that they present to the T helper cell, and T helper cell are presenting to the T cytotoxic cell, which leads to cell mediated immunity. Okay, so this is very important, that skeleton, to understand the cell mediated immunity patterns. Okay, now let's move to our chart to learn this particular. So let's take a look back. MHC class 1 molecules present in all nucleated cells. When cell is infected, they are called altered cell. The altered cell submit the antigen to T cytotoxic cells, which leads to cell mediated immunity. The second possibility with MHC class 2 molecules, that MHC class 2 molecules are present in antigen presenting cells like macrophages, dendritic cell, and B lymphocyte. They submit the antigen to T helper cell, and T helper cell may submit the antigen to the T cytotoxic cells for cell mediated immunity, and T helper cell may submit the antigen to the B lymphocytes for humoral immunity. Now let's talk about cell mediated immunity. Let's talk about the cell mediated immunity. Okay, so here it is, the cell-mediated immunity. So we need to try to understand the pattern of cell-mediated immunity. Okay, so in this, you see here, this is APC, antigen presenting cell. It was given here exogenous antigen. That means microbe is there in the outside the cell, exogenous antigen. When the microbe is present outside the cell, that is engulfed by macrophages. Who is macrophages? Macrophages are nothing but antigen presenting cell. Okay, so that microbe is engulfed, and microbe is processed. You see here that made in the form of fragments. The microbe is processed. Processed microbe is surrounded, that represented on MHC class 2 molecules, and from there it was given to the T helper cell. Okay, so then T helper cell present that to the T cytotoxic cell. That means we need to follow the scenario, whatever that the sequence I explained you to earlier. The same sequence, MHC class 2 molecules containing antigen presenting cell, macrophages are engulfed, processed microbe is presented to the T helper cell, and helper cell is given to the T cytotoxic cell. Okay, that is one story. Then next, that altered cell, ASC. What is altered cell? Altered cell, a nucleated cell, which contain MHC class 1 molecule. MHC class 1 molecule. So that is work with endogenous antigen. That means a cell is there which contain nucleus, our body cell. When microbe is entered inside, we call it as altered cell. So now the cell is presented to the cytotoxic cell, T cytotoxic cell. Okay. So that means T cytotoxic cell getting two possibilities. Via Mhc class 2, that via T helper cell it may get, or directly from Mhc class 1 containing altered cell. Okay, what it will do then? That's the story. T cytotoxic cell differentiated into T memory cells and T effector cells. T effector cells and T memory cells. These T memory cells are going to work in future when the same microbe enters in our body. Then T effector cells. We need to talk about T effector cells, which are also called cytotoxic lymphocyte. T cytotoxic cell, after the activation, when they become effector, we call them as the T cytotoxic T lymphocyte. What they do? T cytotoxic lymphocyte. The T cytotoxic lymphocytes. Okay, little slide. Yeah. The T cytotoxic lymphocyte, what they are doing, you see that the infected cell, that altered cells are infected cell. They are making some holes that we call pores, perforations they make. Okay, so with the help of enzymes called perforins, they make some holes on the infected cell. Okay, so then through the holes, the granzymes are going to be entered, a special kind of enzyme. Granzymes. Okay, so first they made holes, then after the granzymes are entering inside. You can see here, granzymes are entering inside, and they promote a special kind of protein inside called caspases. A special kind of protein called caspases. These caspases, the protein called caspases, promotes these caspases promotes the cell to suicide themselves. That means apoptosis, we use it to say the particular process we can call apoptosis, programmed cell death. Okay, so see the series of sequences. First, they release for perforins, which makes holes. Through the holes, granzymes are entered inside. Granzymes promote the caspases to release. Caspases promote apoptosis. What is apoptosis? Programmed cell death. Apoptosis means programmed cell death. That means our cell is programmed to death, die. So that's it. That is the speciality of that. So that if you understand, if you focus clearly here, this is our body cell. Okay. Okay. So what do you call that particular cell as? A nucleated cell. It is. Okay. So when microbe entered inside, okay, one microbe entered inside, then now this cell is called altered cell. Okay, what this particular, so you see here, thousands and lakhs of microbes are entered inside. What this cell is called now? Altered cell. Okay, then what we did now? Then the altered cell collected some of the microbe, and that microbial fragments are presented over on the surface above on the Mhc class 1 molecule. That means, excuse me, that means giving information to T cytotoxic cell. Okay, these cells, they are giving information. What they do, they'll come and they make holes on our own body cell. They are making holes. You see here, our own body cell, because that is infected cell now. Okay, that is going to be dangerous for us now. So through that particular body cells, that we make holes in them by perforins and we release granzyme. That means what I'm saying, this is our own cell. But we should not entertain that cell, otherwise that will be infected, that will go to the next cell, that will go to the next cell. That's why we are sacrificing. That's why the mode of style of killing of cytotoxic cell is like this. Cytotoxic cell release perforin. Through the perforin, granzymes will enter and they promote the caspases, and we promote the cell to die themselves. That's suicide themselves. What do you call? Apoptosis, programmed cell death. This is the story of that cell-mediated immunity. Two possibilities are there. The two possibilities that I'll show you once in the full diagram. One is like this. From APC to the helper cell to the cytotoxic cell, then effector and memory cells are released. Then another possibility is this one here, ASC to the T cytotoxic cell and the killing. So finally, ultimately, we are going to destroy our own cell which is infected. Infected cell, we don't spare the infected cell. So I request you, after all that the notes you got already, so what you need to do here, that the space I provided here, the space, in that space you need to write the text sentences, that point wise. That means every text sentence is having some related points with this particular diagram. Okay. So that's why you need to read the text sentences and need to write that particular text sentences in the given below cap. First of all, antigen presenting cell. Antigen presenting cell. Antigen presenting cell. Who are called antigen presenting cell? Here it is. We need to go here. So here it is. Antigen presenting cell process the exogenous antigen, this one. And whereas altered cell process the endogenous antigen. Altered cell process the endogenous antigen. Okay. Then the processed antigenic fragments. The processed antigenic fragments are displayed on their APC and ASC. You need to check here that APC and ASC. APC means that Mhc class 2. Then ASC means Mhc class 1. Okay, they are recognized by T cells. Here they are recognized by T helper cell. Here they are recognized by T cytotoxic cell. Okay. Binding of T cells to the APC or ASC cause the production of activated T cell and memory T cell. Here it is a T helper cell should present to the T cytotoxic. Here directly, in case of ASC, directly reach to the T cytotoxic cell. Okay, so that what they said, they differentiate into memory cell and effector cell. The activated T helper cell secrete various kinds of interleukin. Interleukin is a communication language, this one. Okay, so the activated T helper cell which transform and activated T cells to release the T memory cell and T effector cell. In this case, directly it happens. ASC directly present to the T cytotoxic cell, but APC to the helper cell. It will come like this. Okay. Then what happened? The cytotoxic T lymphocyte, that means this one, effector CTL, the cytotoxic T lymphocyte, attached to the perforins, that attach to the perforins and form pores in the cell membrane of the infected cells. You see here that perforin making holes and by releasing perforins. Okay, then grangenes enter the infected cell through this perforation and activate the certain products called, that certain proteins called caspases, which caspases help in the destruction of the infected cell. We call apoptosis. Self-destruction is called apoptosis. Programmed cell death, the meaning of them. Okay. So NK cells will come later. Natural killer cells are similar to their action to Syria. Okay. So this is all about cell-mediated immunity. Whatever here that is there in the diagram. So only thing here, how you look diagram, that is very important. How you are going to look towards the diagram. How you focus on the diagram. When you are talking about APC, should come like this. Okay. Then when you are talking about that ASC, will come directly like this. Okay. So that's it. Whatever activation of the T cytotoxic cell results into T effector cell and T memory cell. T memory cells will do the action in future. T effector cell are going to do the same action is at present condition. So that's it about the cell-mediated immunity. Okay, the cell-mediated immunity. We need to continue with humoral immunity. So for that, let me remind you once that Mhc class 2 molecules are required for that, isn't it? Mhc class 2 molecules are required. The Mhc class 2 molecules present in antigen presenting cells like macrophages, the dendritic cell and B lymphocyte. Okay, B lymphocyte, not only humoral immunity, they're also in this particular action, what we are saying here. Okay, that macrophages, dendritic cell, and B lymphocyte. They submit the antigen to T helper cell. The T helper cell may submit the antigen to T cytotoxic cell for cell mediated. That we are completed. T helper cell may submit the antigen to B lymphocyte. Okay. The interesting thing here that B lymphocyte is there. B lymphocyte is having Mhc class 2 molecule. Okay. So whom they are submitting? They are submitting to the antigen to the T helper cell, and T helper cell again submitting antigen to the B lymphocyte. Okay, in this case, in this particular sequence, not here, in this sequence, B lymphocyte to the helper cell to the B lymphocyte. That means it is coming back again to the same. So that kind of sequence only we are going to watch now. That kind of sequence only here we are going to watch that sequence only here. So let me take a look on this. Let me show you that B lymphocyte. Okay, so this is the cell mediated which we are completed. And now I am moving with this particular story. The same story that for humoral immunity, whatever we are seeing in the theory, here you can see. Now, this is a mature B cell. Now it acts as APC. When microbe entered, they take the microbe inside and make the MHC class 2 molecule. The microbe is processed microbe is attached to the MHC class 2 molecule, and we are presenting this microbe to the T helper cell. Okay, this microbe we are presented to the T helper cell. But what happened? In return, T helper cell again activates that B lymphocyte. Okay, so if we come in the story of macrophages, it will come like this actually. That means macrophages are APCs, which they present it to the T helper cell, and they help T helper cell present to B lymphocyte. Okay, but in this case, that B lymphocytes are also, what do you say them as, antigen presenting cell. So that's why B lymphocyte processed the microbe, and the microbe is given to the T helper cell. Then T helper cell is given again to the B lymphocyte. Now the B lymphocyte will be activated. Activated mature B cells, activated B cell will differentiate into two types of cells. What do you say them as? Plasma cell and memory cells. Okay, plasma cell and memory cell. That means when they communicate with the T helper cell, they differentiate into plasma cell and memory cell. The plasma cell produce the antibodies present. Memory cells are going to produce the antibodies in future when the same microbe enters in our body. Interesting story that to make us to learn about the vaccines even. Okay. So whatever the antibodies are produced here, they start action. Okay. To understand this phenomena, we need to go for a small story. That means with this, simply with this, we can't understand anything here. So that's why to understand this phenomena, we'll go for a small story. That for example, that there are 1,000 microbes are entered in the body. Imagine the microbes are like this. Okay, so that means some, I have to give some shape here. I gave like this. Okay, so some microbes are entered in the body. Okay, some 1,000 microbes are entered in the body. Okay, so these 1,000 microbes are moving in the blood fluids, in the tissue fluids. Okay, so what we do? Macrophages or any APC, macrophages or dendritic cells or that B cells itself having this capacity. They engulf this microbe. That means out of 1,000, imagine some 10 microbes are engulfed. Okay, engulfed and processed. I'm using this word from the beginning, processed. What is the meaning of process? Processed means that making that particular microbe into fragments we call process. Okay, cutting them into pieces, simply we are saying. Okay, so that means this microbe which was taken inside is getting processed, means what we do with them, that macrophages are going to that make that particular microbe into pieces. Okay, so remember this if I say this is the microbe, some shape I given. If I say this is the microbe, among this particular microbe, any one of the part of the microbe can be act as an antigen. That is the difference between the microbe and antigen. Okay. So I'm not showing human being. Please you don't understand that as human being. I'm saying this as a microbe. Okay. For your easy remembrance, I drawn a shape which will look alike. Okay, so that means these are all the microbes. Okay, so among these particular 1,000 microbes are entered in the body. The macrophages are collected some 10 number. Okay, then the processed microbe is presented over on the surface. Okay, and that means among the process, a piece is presented over on the surface. Okay, then let me here. So that we call it as antigen. Okay, that we say it as antigen. What makes this processed microbe to need to be presented to the T helper cell? They observe that information should be given to the B lymphocyte, and B lymphocyte will come and watch what kind of microbe it is. That means imagine this is a lock, what we have to produce, keys. That means modal key preparation is required. Okay, modal key preparation is required. Let me go for a whiteboard to understand all this much better, then after we'll get back. Okay, so that interesting story about the life. So what I said, 1,000 microbes are entered in the body. 1,000 microbes are entered in the body. One of the macrophages are taken the microbe inside. Okay, so then the process, that means we cut them into pieces. Among the processed microbe, that one of the piece is presented over on the surface. Okay, so that this is called antigen now. This is microbe. Okay, so total microbe cannot be antigen. A piece of microbe can be called antigen. Okay, now they present it to the T helper cell. Okay, so T helper cell will react with them. Okay, so will react with them and they give the information to the B lymphocyte. The B lymphocyte has to see this particular microbe. That means antigenic portion. If this is the lock, antigen is called lock, that we need to prepare the key. That key is called antibody. But here the key, how the key we have to prepare, that will take some time. B lymphocyte will take some time to prepare the key against them. That's what we are saying. They differentiate into plasma cell and memory cell. Okay. So in this series, every B lymphocyte will have an antibodies, modal antibodies on their surface. Imagine this is the microbe who is ready. So they need to interact with this particular and should prepare accordingly. Okay, if this particular is circular, so here need to change over. That kind of antibody we need to prepare. Then after we differentiate that into two cells, plasma cells and memory cells, plasma cell and memory cell. So what is the difference here? Which we started preparation of the keys. Memory cells will remember that particular kind of lock. Imagine this is some of the microbe, the well-known microbe. Okay, that well-known microbe Salmonella typha. Let's make it out. This is Salmonella typha. Okay, so that bacteria which entered, we processed, processed microbe is presented over on the T helper cell, that macrophages to the T helper cell, and B lymphocytes observe them, and we process, they differentiated into plasma cell and memory cell. Okay, memory cells will remember the bacteria in future. Now the plasma cells, which B lymphocyte get differentiated, they start producing antibody. Get back to the story that what I said, among the 1,000, we are doing this with 10 number. Okay, we are doing it this 10 number. That means it's just like that in the climax of Robo movie, that the bigger Robo is with large number of robos, that bigger Robo is doing some magic. We brought one Robo, our hero brought one Robo into the laboratory and is testing that. In that way, when 1,000 bacteria entered into the body, with some bacteria we came, we analyzed, we start where that helper cell to the B lymphocyte, we prepared some antibodies. But actually, for the first time, it is very late. We have to collect it, we need to fragment it, and antigenic fragments should be observed by the B lymphocyte. They need to prepare the antibodies accordingly. So all this is going to be a little bit tough and time-taking process. So among them, the 1,000 we have taken only 10, isn't it? The remaining, whatever the remaining, that in the mean process you are going to produce the antibodies. In the mean process you are going to produce that the time you are taking. In the mean period of time, this bacteria will multiply into thousands, lakhs, and crores. That is the reason why we get disease for first time. Why we get disease for first time? That will take some time for the preparation of antibodies because that microbe entered into our body for first time. That microbe entered into our body for first time. That's why we are taken time. That means that microbe, if you compare microbial antigen as a log, that you are looking it for first time, that's why it is difficult to prepare the keys. Whatever we prepare keys, that means that particular antibodies we prepared now, these particular antibodies. How does that antigen microbe is left is seen like this, okay? So these microbes, now these antibodies will go and catch them, will go and catch them. We call it as precipitation or agglutination or complement fixation, various methods are there that will watch. And these antibodies can go and catch wherever they are present in our body, wherever they are present in our body fluids, they go and catch. What happens afterward will go, but we are very near to the vaccination mechanism also. Imagine that I'm talking about Salmonella type 1000 bacteria entered. We said, okay, so now after the exposing to that bacteria, you got memory cells, isn't it? You got memory cell with your key on the surface. Imagine after a period of time, 1,000 the same bacteria entered into our body, the same person entered, that the no need to take sampling, all these things, isn't it? That already that memory cells are present again as the typhoid bacteria. They immediately produce the army that our antibodies and they catch immediately them and they that the destruction will be happens immediately. So that means from this story, what you came to learn, once we get the disease, once we get the disease, okay, this is very important for in this case, in the pandemic situation, once we get the disease, we don't get for second time. We don't get it for second time. Okay, once we get the disease, we don't get it for second time. Why? Why we don't get it for second time? Now we have answer. We prepare we prepare memory cells because of interaction with the microbe for the first time. We prepare memory cells due to interaction. First time it may take time for preparation. In the mean period of time, maybe by microbe may be successful due to the interaction with the microbe for first time that we prepare memory cells. So that's why we act, we react, not we act, we react. We react faster for second time because we already have that information of that particular micro. Okay, so that's why we use it to say that we don't get it for second time. In case of coronavirus, now nowadays we are going to say this, that the particular people in the plasma cells, they'll have some antibodies. How do they have antibodies? How do they have this particular? Because of the microbial infection which they get. Okay, they got the keys of that microbe. Okay, so these memory cells are able to produce more number of antibodies. That's why we don't get it for second time. So here you may get rise a doubt that, sir, for the example you said typhoid, I got it for second time. Some of any of the disease, sir, I got it for second time, third time. I'm getting it continuously, frequently. So what happened to your memory cell? You said memory cells are present. Once we get the disease, we don't get it for second time. So why this happened to me? Why I'm getting the disease again and again? So what is the answer? Answer is there in immunology again. What is the answer? That person's immunity is not up to the level. That person is not able to produce that memory cells. that person is not able to produce adequate number of antibodies even for second time. He is not able to cope up. That means his immune system is weak. Simply we say his immune system is weak. Okay, so that's it. Okay, that means nowadays it became a debate for coronavirus. Once we don't, we'll get it for second time or not. You see the variation. Some people are not getting even for first time. That means our say in the said story we can change a small issue we can change. I said 1000 entered. We are doing the process with 10. No, that in those people who are not even getting for first time, they react faster. That these immune cells are more in number. Their sample itself they are taken whatever entered inside. 1000 is able to kill for first time. Okay, no problem. He is able to kill some 10 100 only. Remaining 900 are multiplied whatever the multiplied, that's why he got it for first time. But that you can see here that for second time he don't get. That means for person to person immune system, reactive immune system activity is going to be that comparatively level wise, stage wise you can see. Okay. So and another thing you remember here that according to the doctors even it is said to be immune system is having a direct relation with the brain, that the stress condition, that means the mental condition of the person immune system is directly connected to the brain. So that means how that stress conditions, anxiety, that fear, these kind of things may the that if infect affect the brain, that may show effect on the immune system. Okay, so that person fearless, stress-free, that without anxiety, his immune system work much better than the normal person. Those people who feel stress, anxiety, and fear. It was a proven fact. It was a proven fact. Actually, that every activity with our body, that how calm we are, that based on that only. That means I'll go with an example. When you are in stress, when you're anxiety, when you're in fear, or when you're under pressure, when you are under pressure, that can I say like that your digestion is not proper? Yeah, you may felt over this particular condition under the examination pressure. You don't feel any hunger. That means why? That the digestion is not completed. That means here that everything is related to the brain. So in that way, immune system is also related to the brain. Okay, let's get back what antibodies will do. That we need to come through. That at least we came to understand the second time we don't get. The disease, that is because of the memory cells which we develop in both cases, in B lymphocyte as well as in T lymphocyte also. I just shown example in the form of B lymphocyte to you. Okay. Now then, what these antibodies do? That's the question now. If you have an idea that antibodies who are released are going to kill the microbe, going to destroy the microbe, act like a Superman or Spider-Man, that's wrong. Antibody is a protein, just antibody is just a protein. What it will do? It will bind with the microbe, as I said to you. This is the microbe in the given example. So that microbe, this is the antigenic portion of the microbe. So our antibody will go and hold that particular antigenic portion. Okay. So that when they do like that, what do you call it as? We may say that that my antibody, this is antibody. Okay. Wait a minute. Excuse me. This is antibody and this is the microbe. Okay, where antibody binds, that portion we call antigen. So this antigen and antibody complex is what do you say that will happen over in our body. Okay, so microbe is rotating in the body fluid. Our said antibody will go and bind with that antigenic portion of the microbe. That only you are looking here in the diagram. We call it as neutralization. When antigen and antibody complex is formed, we call it as neutralization. In both cases, virus or microbe, you can see here. Let us see in microscope to understand much better. I am showing a little bit of that zoom, making this area zoom. Okay, so that you can see here that virus, this is the virus. It is having many antigenic binding sites. where antibody go and hold them, neutralize that particular microbe. And here also the same, bacteria in this place, in this place, in this place, antigenic fragments are there and will go and bind. What do you call it as? Neutralization. We can say it as neutralization. Okay. Next, wait, let me erase this. I'm not able to erase this. I think struck over. Okay, so that's why I'm doing it back again. Yeah. Next, the binding of antigen with antibody seems to be in the form of the agglutination, clumping, we can say. Sometimes it seems to be clumping, not always. Sometimes it may see in the form of clumping. Sometimes it may show in the form of precipitation. So precipitation or agglutination or nothing you can see here. That means when a bind antigen and antibody complex, not a rule like that. For every time that microbe should form, the complex should form something. That sometimes they form agglutination, sometimes they form precipitation. For example, here the precipitation. Imagine this is a syphilis bacteria. With syphilis bacteria, antigen antibody complex form the precipitate. Okay, powder like a small substance we see. So that, what do you say, that antigen and antibody complex? Okay, so what we do with this? Okay, antigen and antibody went and bind with the antigen and form the complex. What we do with this? What we do, here it is, what we do. Phagocytosis. In our body, neutrophils are there, isn't it? Macrophages, neutrophil. What they do? Along with the complex, antibody and antigen, the whole complex is going to be engulfed over. The whole complex is going to be engulfed. Okay, you need to understand this actually. Antibody is not killing, just going and holding. Okay, so that holding that particular microbe, and later work is done by neutrophils. Okay, so the total process, what do you say, with the antibodies or roaming or moving in the body fluids, we call such type of immunity as humoral immunity. Okay, so let me show you a glance look on this particular area. That, let me show you once again, that this is the mature B cell, that when the B cell is acting as a APC at present, that they interact with the antigen microbe, and that they inform to the T helper cell, that with the help of Mhc Class 2 molecules, and then the T helper cell again activates the B lymphocyte for the differentiation of the plasma cell into and memory cell. These plasma cells, you see here, endoplasmic reticulum activity is going on because they have to produce antibodies, protein synthesis is going on, and that particular proteins, antibodies are prepared, and prepared antibodies released inside in the body and will search for the roaming microbes in the body, and they form either the neutralization may happen, or agglutination, or precipitation. Whatever happen, finally that leads to the phagocytosis. The total complex will be removed by phagocytosis. Okay, so this is the story of humoral immunity. Let us read the same in the textbook, whatever we learned. The mechanism of humoral immunity, that after the maturation in the secondary lymphoid organ, the free antigens bind to the FAB, and we need to look forward toward the antibody also. But till the time you remember, this portion is called FC and this portion is called FAB. What is AB? Antigen binding site. The fragment antigen binding site. This is the antigen binding site. This is the constant region. Okay, so that FAB end, the free antigens bind to the FAB end. This is the thing they are saying. The free antigens bind to the FAB end, and on the surface of mature B cell, they engulf and process the antigen. Then they display the antigenic fragments on their membrane with the help of class 2 MHC molecule. Means at the time they are acting as APC, antigen presenting cell. Then T helper cell recognize them and interact with the antigen of MHC2 complex molecule. You can see whatever the theory visualized here. Okay, and release a type of interleukin. Interleukin is a language between the cells. Okay, so then we stimulate the B cells to proliferate. Again, they do They release the interleukin and they stimulate the B cells to proliferate and differentiate into plasma cells and memory cells. The plasma cell release specific antibodies into the plasma or extracellular fluid. These antibodies help in opsonizing and immobilizing. What is immobilizing? That you have seen, neutralize. Opsonization is also similarly like. That means the opsonization, making bacteria more susceptible for phagocytosis. We can say opsonization. That same. That means here holding the microbe. That with the help of that they hold the microbe. Okay, so that means here if you want to hit a person A, okay, B and C will come and catch this person. You are the D, you can hit him clearly. Okay, so I'm not promoting any kind of these violent action. Just example I'm saying. If you want to beat A, so we are taking the help of B and C. B and C that caught him right and left side, and you are the D, you can hit him directly. So what do you say this as? That B and C, what these B and C are doing? Opsonization they are doing. So that same we are saying antibody that will catch that microbe and that neutrophils can come and hit them. So that what you call opsonization. By definition wise, we can say making bacteria more susceptible for phagocytosis. Okay, immobilizing the bacteria, neutralizing, opsonizing, that cross-linking of antigens leading to sometimes either in the form of agglutination or unsoluble, the agglutination of unsoluble or precipitation of soluble. That means the complex if dissolved, that we can see in the form of precipitation. If not dissolved, that can be seen in the form of agglutination. They also activate the phagocytes and complement system. That what is complement system will come in future. That phagocyte activation, or at least you can see here. That the phagocyte activation you can see here. So this is the phagocytic activation, and that antigen and antibody complex is engulfed. Okay, this complement system I'll go with later. That means when the series will come, that I'll show this diagram for you back again. Complement system. So that's it. It is all about the humoral immunity and cell-mediated immunity. Humoral and cell-mediated immunity. Yes, a big area. That means the immunology is a complex area. With our limits, we are going to learn this particular topic with our limits. Okay. So finally, you may get a question like, what is possible with the B lymphocytes, humoral or cell-mediated? Likewise, a question come over. But when learning for first time, we need to go with the complete topic in detail. So in that way, we are moving. Okay. This is the diagram which we are looking here. That is antibody, basic antibody, how it seems to be. That you can see in the diagram. So this is antibody. Each antibody is made of the 4 polypeptide chains. First of all, you remember antibody is protein. Like any enzyme, like any other substances, any proteins you are doing, like you learned in your earlier classes about the protein synthesis, here also the same. Similar kind of protein synthesis will happen. Okay, so that antibody is a protein. It's not a cell, or it's not any kind of organism or any kind of active component. It is a protein. Okay, it's just like hormone or just like a protein hormone or just like an enzyme. Okay, that means they are proteins. I'm saying that you need to recognize that that is protein. It is made up of 2 heavy polypeptide. Protein means definitely we'll find heavy polypeptide chains and light polypeptide chains. So 4 polypeptide chains, in that 2 heavy polypeptide chain and 2 are light polypeptide chain which bind with disulfide bonds that like this. Then component wise there are 2 portions here that in every antibody. FC end. What is FC? FC means fragment constant region. That fragment, let me write here, the fragment constant region. It's there in the name, constant. You don't change for any antibody, for any kind of microbe. Changing portion is this area, the fragment constant region. Okay, then actually the FC end may attach to some B cells, as you have seen in the earlier diagram. With the help of FC end, it is attached to the B cells. isn't it? Okay, or it may attach to sometimes to the mast cells or B cells. They get attached to that all the time. We can't say that these antibodies will attach only to the cell membrane. We'll find free antibodies that will rotate in the body fluid. That means we can see that antibodies are of two types. That some are going to attach to cells like B cells and mast cells, and some are going to be free. Okay. Next, fragment antibody by antigen binding site. A, B. What is A? Antigen. B, binding site. Okay, fragment antigen binding site. Till the time we discuss about this only. That means accordance to the antigen, accordance to the microbial portion, this area is going to be changed over to, for that change only it will take some time. For that only it will take some time. So what do you say it as fragment antigen binding site? We call it as fragment antigen binding site. Okay. So then here only we'll complete the definition. If this is antigen, that means a microbe which is going to bind. This is the portion. Okay, so I'm saying it as microbial antigen. Okay, so this is the portion where it can bind with antibody. I'm writing here antigen. So this is the portion which it can bind with antibody. So the portion of antigen which binds with an antibody is called, the portion of antigen which binds with antibody is called epitope. The portion of antibody which binds with antigen is called paratope. You will be confused, that's why I'm showing both at one place. So paratope, you need to remember like paratope belongs to antibody. What it will do, it bind with antigen. Okay, epitope belongs to antigen. What it will do, it will bind with antibody at FAB. Okay, so that means two definitions you need to learn here. Epitope is a portion of antigen which binds with antibody. The paratope is a portion of antibody which binds with antigen. Okay, so that means epitope of antigen binds with the paratope of antibody. Okay, so that's it about the story of this particular antibody with 4 polypeptide chain. In the 2 are heavy polypeptide chain, 2 are light polypeptide chain, and the 2 portions are present, FC end and FAB end. FC end is a constant region. It may attach to the B lymphocyte or plasma cell, or it may be free. Okay, FAB end is the portion which modifies according to the microbial entry. That what do you say of ABN? Antigen binding site. The portion of antigen binding site, the portion of antibody which can bind with the antigen, we call paratope. Okay, then there we have and antigens. We used to say it as immunoglobulins. The name for antigen is immunoglobulin. Immunoglobulin. In general terms, we call the immunoglobulins as Ig. Ig. What is Ig? Immunoglobulin. Okay, so there are 5 types of immunoglobulins are present. We use it to say simply as Ig. Okay, there are 5 types of immunoglobulins are present. IgG, IgM, IgA, IgD, IgE. There are 5 types of antibodies. Immunoglobulins are 5 types. IgG, IgM, IgA, IgD, and IgE. We'll close the session today by showing these 5 types of antibodies. See this. These are the types of immunoglobulins. In that the first one is Igd antibodies. Some information we require here, that I gathered more than the required information. Just for the knowledge, you have to remember it. Just for once, you need to take over. Then some basic questions are coming from here, but no matter, I gathered little bit more than requirement. Igd antibodies. You see the Igd antibodies in the picture. These are the Igd antibodies. Igd antibodies do not activate the complement system and cannot cross the placenta. Properties they are saying. They do not actually antibodies. We use it to say they activate a complement system, but these antibodies do not activate the complement system. They cannot come across the placenta. That means mother to fetus, they cannot travel, that they are saying. They are mostly found on the surfaces of B cell, probably functioning as a antigen receptors, mostly found on the surface of B cell. Earlier, we have seen some antibodies on the B cell, isn't it? They are nothing but IgD. Okay, the probably functioning as an antigen receptor that help initiate them differentiation of the B cell into plasma cell and memory B cell. That means earlier story we learned over that be about these antibodies only, IgD. Okay, IgM, pentamers these are. You see here, the 5 antibodies in one place. That what do you say? The pentamer or polymer, that IgM, are the first circulating antibodies to appear in response to the antigen. So IgM are the first circulating antibodies to appear in response to an initial exposure to an antigen. They concentrate in the blood, then declines rapidly. Thus, the presence of IgM usually indicates a current infection. Remember this line, that the presence of IgM usually indicates a current infection. You may heard over in case of COVID cases, we are doing antibody testing. Okay, so that means you see this line that will correlate. Thus, the presence of IgM usually indicates a current infection. Whether you got infection or not, we can say any infection, but nowadays the famous one is COVID. So that means if you are having more IgM antibodies, you may have infection in that way. Okay, their concentration in the blood then declines rapidly, and the presence of IgM usually indicates current infection. IgM consists of 5 Y-shaped monomers arranged in a pentagonal structure that you can see in the diagram. The numerous antigen binding sites make it very effective in agglutinating antigens and in reactions involving complement. IgM is too large to cross the placenta because it's a bigger one, you see, and does not confer the maternal immunity. Okay, next, IgA. IgA is produced by cells in the mucous membranes. The main function of IgA is to prevent the attachment of viruses and bacteria to epithelial surfaces. The main function of IgA is to prevent that virus or bacteria, when it enters, what it will do? It will attach to the cell surface and will try to enter inside. That IgA can stop, prevent the attachment of the viruses and bacteria to epithelial surfaces. IgA is also found in many body secretions such as saliva, perspiration, and tears. Its presence in the first milk produced helps to protect the infant from gastrointestinal infection. That means colostrum we are talking, mother's milk. Next, Igg antibodies is the most abundant of the circulating antibodies. It readily crosses the walls of blood vessels and enters tissue fluids. It can come across the tissue fluid. Igg also crosses the placenta and confers the passive immunity on the fetus. Igg is the most abundant of the circulating antibodies. It readily crosses the walls of blood vessel. Keep this point, remember this point here, that and enters tissue fluid. Igg also crosses the placenta and confer the passive immunity on the fetus. Okay, so this is also going to be one of the reasons for erythroblastosis fetalis. Please recall the erythroblastosis. Igg can come across the placenta. Igg also crosses the placenta, confers the passive immunity on the fetus. It protects against bacteria, viruses, toxins in the blood and lymph, and triggers action of the complement system. Next, finally, antibodies E, IgE, is slightly larger than IgG, okay, and represent only a small fraction of the antibodies in the blood. That the tails attached to the mast cells and basophil. I said over some of the antibodies will attach to the basophils and mast cells when that these are IgE. When triggered by an antigen, cause the cells to release that inflammatory mediators like histamine and other chemicals and cause an allergic reaction. That is all about IgE antibodies. So the 5 types of antibodies with their different roles and different activities: IgD, IgM, and IgA, IgG, and IgE. Okay, so then